Clinical Microbiology, Introduction to Clinical Microbiology and Lab Safety. So when we talk about medical laboratory technician as a profession, we know that the medical laboratory technician diagnoses disease, and they play a pivotal role in the healthcare team. They work in the lab, gather specimens, put those specimens on analyzers to report results to the ordering physicians. There's different areas in the clinical lab and different ways that we can diagnose disease. In clinical chemistry, we evaluate analytes from blood samples to identify which cells or organs may be injured. Now, that's not a comprehensive definition, but in general, you're going to take blood specimens and see which chemicals are present in the blood. Molecular diagnostics, you evaluate genetic material to identify the presence of select genes or errors within a gene. Immunology or serology, you use antibodies or you evaluate the presence of antibodies in a blood sample to detect disease. Urinalysis, you're going to look at the urine for abnormal constituents, so abnormal components to the urine, to determine a cause of disease or provide further evidence of disease. Hematology, you're going to look at the shape of the blood cells and the number of blood cells and blood components to determine whether there are abnormalities that could cause disease. Cytology or histology, you can evaluate cells in cytology or tissue samples in histology for the presence of disease. Microbiology, you evaluate specimens for the presence of pathogens and attempt to isolate the pathogen to determine its identity and susceptibility to drug treatments. So what are pathogens? Pathogens are disease-causing microorganisms. So if you suspect that a patient has an infection, that patient is going to get swabbed, and that swab is going to come down to the microbiology lab. And then it's the role of the clinical microbiologist to work up that swab to see what's actually causing the infection. Disease-causing microorganisms could include viruses. Viruses are acellular, which means they are not considered cells. All they are is some genetic material wrapped in a protein coat. Viruses are highly transmissible, and the naked viruses, the ones that don't have a capsule, are pretty easy to kill. So, deactivate, sorry. So viruses are very widespread. A lot of times with viral infections, we don't actually know what virus. We don't actually go to the level of diagnosing the virus unless we have a molecular test that can test for some of the more common viral particles. Bacteria are going to be probably the most commonly encountered organism in the clinical microbiology lab. Bacteria are prokaryotes. Prokaryotes means that they do not have a nucleus, and they have peptidoglycan in their cell walls for the most part. There are a few exceptions to that rule, but bacteria are going to be simple unicellular organisms. Fungi include the yeasts and the molds. Fungi are eukaryotic organisms, which mean that the cells do have a nucleus. Protozoa are also eukaryotic organisms, but protozoa are going to be single-celled and they're animal-like, so they have very similar structures as animal cells. Helminths are not microscopic themselves, but we cover them in the field of microbiology because their eggs are. So helminths are the worms. Worms are large multicellular organisms that lay small eggs that we can see under the microscope. So the roles of the clinical microbiologist to culture organisms from specimens. It gives you a snapshot of the specimen. So when we get a swab from a patient, we're going to then swab it onto an agar plate and try to culture it. And when we culture it, the goal is to grow enough of it so that we can figure out what's causing the problem. Classification and identification of organisms, so the possible cause of disease. Right away when we get a specimen from a patient, we can start to evaluate certain features of the microorganism, especially if it comes from a sterile source. So, for example, if we get a cerebral spinal fluid specimen, and we spin it down, so we centrifuge it, and all of the clumpy bacteria stuff goes to the bottom of the tube. We can then take a sample of the bacteria that fell to the bottom of the tube, put it on a slide, and gram stain it directly, and we can start to evaluate what cells were found in that sample. If we see certain types of cells, then we can give clues to the doctor as to what could be causing the infection. Prediction and interpretation of susceptibility. So prediction and interpretation of susceptibility is talking about the susceptibility to antibiotics. So whether or not the antibiotic is going to be able to kill the microorganism that we're trying to get rid of. Knowing what the microorganism is susceptible to will improve treatment. Bacterial relationships to isolation. So growth requirements of bacteria allows microbiologists to select the correct medium for the primary culture. So depending on what source the swab came from or what source the infection is found in, we're going to plate it on different types of media, which we'll go into in detail later on in the course. And it increases the likelihood of pathogen isolation. So if we think about like a stool sample, how many different species of microorganisms are going to be found in stool? Lots. So how do we isolate or separate out the one that's actually causing the problem from the good guys or from the neutral guys, the guys that we don't really care about? So that all has to do with selective and differential media, which we will get into in this course. The steps in the bacterial identification determine the staining characteristics based on cell wall structure. In other words, do like a gram stain, or we can do an acid-fast stain if we suspect mycobacterium. Organism size and organism shape. So if we know that it's super small and it's circular, we would call it a cocci. If it's elongated and it's more rod-shaped, we would call it a bacillus, and so forth. And then we can observe metabolic biochemical reactions. Now, all of these steps are the fundamentals of microbiology. Putting bacteria into media, seeing if the bacteria can or cannot eat a certain nutrient, detecting whether it can or cannot eat that certain nutrient by using color-changing media or pH indicators in the media, and then being able to report out those results. If we know one bacterium can eat glucose but not lactose, and our patient specimen can eat glucose but not lactose, then we can kind of say, oh, well, our patient specimen eats the same things as this known organism, and then we can deduce the identity of the microorganism. And we always correlate that with the gram stain, with what it looks like on a plate, and we also correlate it with the patient's signs and symptoms. However, today, a lot of the testing is going to molecular testing. So instead of doing these culture methods, which take a long period of time to be able to isolate and identify, we do a lot of PCR or polymerase chain reaction, a lot of gene amplification or gene probes, and we will get into that in the molecular diagnostics course in this program. But a little bit later in this course, we're going to talk about some molecular diagnostic techniques. So that's kind of the way of the future, but in order to pass our qualifying exam, you gotta learn the basics. Also, if we ever don't have that technology, like let's say you want to travel abroad to a part of the world that doesn't have advanced molecular diagnostics, you have to be able to know how to culture all these microorganisms. Or if you're working right here in the United States and those molecular diagnostic tools break, you got to go back to the basics and be able to culture by hand and be able to interpret those results. So we, in this class, will be focusing heavily on the basic culture techniques to be able to identify using the standard practices. So more duties of the clinical microbiologist: lab safety and regulatory compliance. So when you work in clinical microbiology, when you become a medical laboratory technician and you're working in the clinical lab, your role in the clinical lab includes lab safety and regulatory compliance. So lab safety, obviously we want a safe environment for everybody. Regulatory compliance, making sure that your lab area is in compliance with all sorts of governing agencies so that we report out results that do not have errors in them. So keeping a safe environment, making sure all of our media is up to date and not expired, that we are cleaning appropriately so that there's no contamination in our cultures, and all of those types of practices we will cover extensively in this course. Then we have pre-analytical, analytical, and post-analytical evaluation. In every component of this course, we're going to come back to these three basic areas. It is extremely important that you write down what these are and be able to answer questions about what they are on your qualifying exam. Pre-analytical evaluation has to do with evaluating the specimen before we actually start testing. Producing a specimen or getting a specimen and all of the specimen workup that happens before we actually analyze it, before we actually put it on an analyzer or actually go ahead and test it. Pre-analytical evaluation includes like patient identifiers. You always want to have 2+ patient identifiers on a specimen. So you can't just say, oh, this is the name. And if the name is like Bob Johnson, that's the most common name out there. You know, if there's 2 Bob Johnsons, you could screw up or mix up those patients. So we want to have at least one other identifier on there. It's preferable to have at least 3 patient identifiers on a specimen. Collecting the right specimen at the right time, collecting the specimen in the correct container, transporting it appropriately, processing it once it gets down to the lab before we put it on the analyzer. So all of those components are considered pre-analytical. That all happens before we actually perform the tests. Analytical evaluation is actually performing the tests. So that would be streaking the bacteria out on the media for isolation, looking at the colonies, QA, QC, troubleshooting. So quality assurance, quality control, any type of troubleshooting you have to do, all of that is included in the testing phase. And then when you're done testing it, you have to interpret those results and report those results up to the ordering physician. So that's all included in post-analytical evaluation. So if we do have an error in the test system, this is when that error would get caught. And this is where most of the critical thinking happens. So thinking about what happened and how to fix it and how to report the correct results, making sure that the results make sense before we send them up to the floor. So post-analytical evaluation is everything that happens after the test is done and kind of interpreting and making sense of those results.